So we'll have a little bit of immune system towards the beginning there.
If you wish you had a roast beef and a beef and cheddar right now, you should know Arby's wishes they had $6.
Yeah, they have a mix and match right now.
Maybe you two could work something out.
That's what you need to know.
Arby's, we have the meats.
It is time. We have arrived at the final conflict, the Battle Royale. A fight to the, well, hopefully not death, but a big fight anyway. It only seems fitting that we spend this, our last episode of Anatomy and Physiology, talking about one of your body's last-ditch efforts to defend itself at all costs. This is what happens when all the other fail-safes have failed. Your skin and mucous membranes did what they could as physical barriers against infection, and your humoral immune response cranked out antibodies in an effort to keep your interstitial spaces healthy. But when those systems weren't enough, your cells themselves were breached, and pathogens and abnormalities began to run amok where antibodies could not get to them. Now it becomes the business of your cell-mediated or cellular immune response. And that's where stuff gets real, where cell fights cell, and where heroes look like T lymphocytes. These lymphocytes, known on the streets as T cells, go after body cells that have been hijacked by things like viruses or bacteria or become cancerous. T cells cause inflammation, activate macrophages, get other T cells fired up, and generally regulate much of the immune response. Which is important because of all the ways in this course that we have described how you could die, two that we have neglected to mention are what happens if your body completely fails to protect itself, and what happens if your immune system goes rogue and attacks your own healthy cells. But even when they're functioning well, some of your immune cells are careening around your body like miniature biological versions of Mad Max war boys, amped up on signaling chemicals, scouring the terrain for hostiles, and covered from top to bottom in the dismembered parts of the enemies that they have vanquished. They're out to protect all of the tissues and organ systems that we've been talking about for the past 46 weeks, and these guys play for keeps. If there are cells in your body that look and act like they're from some post-apocalyptic hellscape, it's gotta be the cells in your immune system. Aside from the fact that they go around literally eating their enemies and have names like natural killers, some of these cells are dressed for the part too. Specifically, they go around wearing parts of the organisms they've killed so others can see them. Sounds a little bit messed up, but we're talking life and death here. The stakes are high. And this kind of behavior occurs both in your innate immune response and your acquired response. We've already talked about how in the innate response, when a phagocyte sees a suspicious character, it engulfs it and kills it, right? But what we didn't get into before is that during its attack, the phagocyte actually breaks the pathogen into tons of tiny molecules and then proudly displays those broken bits in grooved proteins on its outer membrane. These proteins are called major histocompatibility complexes, or MHCs, and they're a lot like how Vlad the Impaler decorated his front yard with the bodies of his skewered enemies, or how a battle-crazed warrior might show off a necklace made of knuckle bones. Because cells from both your innate and adaptive branches do this grisly accessorizing like it's their job, they are referred to as professional antigen-presenting cells. Which might make you think, is there an amateur version of an antigen-presenting cell? And yeah, there kinda is. Every nucleated cell in your body, which means all of your cells except for red blood cells, have one kind of MHC protein on their surface called class 1 MHC. MHC1 proteins present short chains of amino acids that are based on endogenous proteins, that is, proteins synthesized inside that cell. So if a particular cell is healthy, the antigens on its MHC1 tell roving immune cells that everything is okay inside, nothing to see here. But if the cell is, say, cancerous and it's making abnormal proteins, then it'll fix bits of those proteins to its MHC, which alerts immune cells that there's a problem inside and basically asks to be killed. Now, your immune-related cells, like macrophages, dendritic cells, and B cells, wear class 2 MHC proteins on their outsides. These are the professionals. Class 2 MHC proteins bind to fragments of exogenous antigens, like a virus that's been engulfed, broken up, and displayed to get the attention of other cells. And this is how MHCs are totally essential to the cellular immune response. Because the heroes of your cellular defenses, the T cells, can't actually detect whole antigens. They can only recognize them when they're all diced up and decorating an antigen-presenting cell. T cells are made in the bone marrow, but they mature in the thymus, the lymphoid gland that sits on top of your heart, and which is actually what the T in T cell stands for. And you have several different kinds of T cells, but the two you really have to know about are helper Ts and cytotoxic cells. Helper Ts themselves can't kill, kill, but they can activate cells that do, and they help call the shots for the whole adaptive immune response. Meanwhile, cytotoxic cells are the ones that actually do the killing of the cells gone bad. Now, much like how a naive B cell carries antibodies for one specific antigen, a naive helper T cell has receptors that will only bind to one specific combination of a class II MHC and a particular antigen. If that match is right, the helper T bonds to the MHC antigen bit and it gets activated. Then, just like with the B cells we talked about last time, the helper T starts copying itself like crazy, making a few memory T cells as well, which remember that particular antigen should it meet one again in the future. And it also produces a whole mess of effector T cells, mostly more helper Ts, but also some regulatory T cells that I will get to in a minute. But the main thing the helper T cells do is raise the alarm that tells other immune cells that there is a problem. And they do this by releasing a cocktail of chemical messengers called cytokines. When a cytokine enters another helper T, that cell usually starts dividing, making more memory T cells and more helper Ts, which release more cytokines that keep boosting the signal. And some of those cytokines also go on to help activate the cytotoxic T cells. You know that macrophages from the innate system just sort of roll up and swallow pathogens whole, but cytotoxic T cells do their killing a little differently. They roam the blood and lymph looking for hijacked amateur body cells that are asking to be killed. Basically, these infected cells are already dying, so they've digested some of their invaders' proteins and stuck them on some of their class I MHCs, effectively waving a surrender flag made of fragments of the very virus or cancer that is destroying them. If a cytotoxic T cell with the right receptor floats by, it binds to the antigen MHC combination and moves in for a mercy killing. It does this by releasing special enzymes that punch holes in the cell's membrane or otherwise trigger apoptosis, killing both the cell and whatever is inside of it. Then the cytotoxic cell just detaches and continues to run down other prey. So by now it should be pretty obvious that without T cells, there basically is no adaptive immune response, and it really all comes back to the helper Ts, which is why immunodeficiencies can be so deadly. AIDS, for example, is caused by the human immunodeficiency virus that specifically attacks helper T cells, and without the helper Ts, there wouldn't be much of a humoral response either, because the cytokines that come screaming out of the helpers not only activate other T cells, but they also finish the training of the B cells. The fact is, most of your so-called naive B cells don't get fully activated and become memory or effector cells when they first bind to an antigen. And there's a good reason for that. Since antibody receptors are generated randomly, you might wind up with B cells that could bind to your own healthy proteins, like, say, your growth hormone. So once a B cell interacts with a substance, whether it's growth hormone or some dangerous bacterium, it still needs to bind to it, engulf it, and present some fragments of it on its surface. But then it'll stop to await inspection. It pauses until the right helper T cell comes by to check out its presentation. If the T cell binds to the presented fragment, then it releases cytokines which fully activate the B cell, and suddenly you got antibodies going everywhere. But if it doesn't, then the B cell just goes about its business and doesn't trigger an immune response. This check and balance between Bs and Ts is an important safeguard against your immune system becoming too good at its job, which is a very real risk. A hyperactive immune system can cause mayhem by losing its ability to distinguish enemy from self as it turns on your own body. Your regulatory T cells, another type of effector, help prevent this by releasing inhibiting cytokines that tell other immune cells to stand down once the initial threat has been handled. Without that regulation, the body might start cranking out too many antibodies and cytotoxic cells that could damage or destroy its own tissues. This dangerous confusion is what causes many autoimmune diseases, like multiple sclerosis, which eats away at the myelin sheaths around neurons, or type 1 diabetes, which tears up the pancreatic cells that make insulin. So the takeaway here is that your immune system is usually really good at its job, which is to kill stuff in the name of keeping you alive. And you really don't want it to go rogue on you, because if there's one thing you should have learned in the past year with us, it's that your body is both resilient and fragile, and it survives only when the sum of its many complicated parts stays balanced and works together. And that is the glorious wonder of you. As we wrapped up our tour of the immune system today, you learned how the cellular immune response uses helper, cytotoxic, and regulatory T cells to attack body cells compromised by pathogens. We looked at how cytokines activate B and T cells, and what happens if your immune system goes rogue and starts causing autoimmune trouble. Thank you to our headmaster of learning, Linnea Boyev, and thank you to all of our Patreon patrons whose monthly contributions help make Crash Course possible, not only for themselves, but for everybody.
At least bringing some of those terms that have to do with our T cells that we discussed before, bringing those kind of back to your consciousness and thinking about, okay, so we have different types of T cells, cytotoxic, helper, we even mentioned regulatory T cells and things like that, memory T cells, and so how each one of those contribute to that third line of defense in some way, right? So we got to think about, okay, what does the cell do? What is, if it produces molecules that are going to be important in defense, what are those molecules? So like when our helper T cells are producing those cytokines that are stimulating other types of cells. Our cytotoxic T cells are producing things like our perforins and our granzymes, et cetera, that can kill off other cells, things like that. Okay? So, before we move on to respiratory system, any other questions with immune system? Okay. So let's take a look at another system, hopefully one that you find easier and somewhat simple, for various reasons. Number one, as we had talked about before and as we had seen in lab, right, the respiratory system is a fairly compact system, so fewer parts, and we're back to a system where we have larger organs that we can point out, that we can identify, etc., and talk about how they contribute to the system. Like usual, we'll do our big picture about what the system does, and then look at the different organs and how they contribute, and then we'll get down to more of the physiology, right? So what it does in order to support the body itself. So that's going to be key. So some of those functions are going to be pretty simple, things that you would recognize and that you've known all along the way. But we may just need to understand them a little bit better in terms of how they do that, right? We've already measured lung volumes. We know that air goes into and out of the lungs, but we need to understand how that happens, right? So we just like, oh, I breathe in, and when I want to breathe in, I do it. But how does that work, right? And we may have a basic understanding of some of the muscles involved. Like, what are some of the muscles involved that make the lungs inflate and deflate? Diaphragm, that's a big one. That's the primary one. And then we have some of our intercostals, right? The external and internal intercostals. Those are going to contribute as well. So we know the muscles involved, but again, how does that get air to move into and out of the lungs? Muscles just move, but they're not grabbing onto air molecules and pulling them in or pushing them out, right? So we have to understand how that process works too. And as we had introduced this system, we said that luckily, a lot of the ways that this system functions is similar to how the cardiovascular system functions. Again, we're going to be talking about pressure, moving volumes of things. We're talking about tubes, thick tubes that can change diameter, that can affect flow and pressure and things like that. Thinner structures that can allow exchange across their walls. So a lot of the groundwork that we laid in cardiovascular system we'll see here as well. Okay? All right. So the main reason, though, that we have this system overall is that your cells and their needs are driving it, right? So we have billions of cells that make up the different tissues, make up the different organs, make up the organ systems, make up you. And each one has their cellular metabolism, their different requirements that are very similar to what your requirements are, right? So in the beginning of 201 or in 181, you talk about some of the needs of those cells. They need to be able to get rid of waste products. They need to be able to respond to the external environment and control the internal environment. They need to get oxygen. They need to get nutrients. They need to get fluids. All of these things that we talk about you as an individual organism and your needs also scale down to the individual living cell and its needs, right? And so you need oxygen, and you're like, well, that's why I have a respiratory system, because I need oxygen. If I don't have oxygen, then I'm going to die. Very true, but why? Like, what is the specific physiological need that we have for this gas, right? Why is it not nitrogen, which is most of what's making up the atmosphere? So in fact, again, that harkens back to that general biology that we learned in terms of what's going on inside the cells. One of the main ways that they produce ATP, that all-important energy molecule that fuels the activity of the cell and things like that, is through aerobic respiration, right? So you're back to powerhouse of the cell and all that type of thing, right? It ends up being the final electron acceptor. We have that electron transport chain, right? We're not going to go through all the details about that. But at the very end of that stepwise process, you have oxygen that's going to be accepting that electron. at its lowest level. And so we have to have oxygen for that cellular respiration. And so we're actually seeing a tie between aerobic respiration, what's going on inside the cell, and this whole system of the body called the respiratory system that's supporting that. So the cellular needs are driving the need for this whole system of the body here. And of course, carbon dioxide, which that should be a subscript, it's not oxygen squared per se, but carbon dioxide is a byproduct of that process as well that can accumulate that we need to get rid of. So again, driven by what's going on down in the cell itself. All right, now there are a few other functions for respiratory system as well, right? Special senses are going to be involved. So olfaction, our chemoreceptors associated with picking up molecules in the air that we're bringing in and pushing out. That's going to be a function. And then speech. We've looked at the larynx and how we have those specialized vocal folds in there. And as the air passes through it, we move those vocal folds. We can produce sound, which allows us to communicate or sing songs or whatever we want to do. All right. So overall functions, yes. Now overall structure. So like when we were doing cardiovascular system, we're like, okay, what are the parts that make it up? We have the heart, we have the arteries, you got the veins, the capillaries, and things like that. And where they set into these different kind of circuits at that point, zones at this point for respiratory system. So we kind of have two different zones, and sometimes the respiratory system is also separated into like an upper and lower respiratory tract. We talked about that a little bit in lab. For the most part, the upper respiratory tract, as well as some of our lower, thicker parts of the lower respiratory tract, is part of our conducting zone. So conducting is just about moving air from one place to another, like some of our large blood vessels. It's not going to be exchanged across the wall. It's just about moving it from one place to another. So our conducting zone is getting it to and from the site where exchange is going to occur, where we're going to exchange oxygen and carbon dioxide. So the bigger structures with the thicker walls are going to be part of the conducting zone, right? Nose, nasal cavity, pharynx, back of the throat. Technically, we get to the larynx where we're separating our air from our food and our drink, and then down to the trachea as well. And even some of our bronchi. Bronchi are big structures, thick wall, cartilaginous rings. There's not going to be things going across the airways there. So all of that's conducting, just getting it in and out. But as we work our way down, as we've seen in the models, we get to smaller and smaller and thinner branches. That's when we get down to the respiratory zone. So respiratory is going to be about those individual respiratory gases that we talked about, oxygen and carbon dioxide, and allowing them to exchange across the walls. So we have to get down to small enough and thin enough structures where those gases can exchange with something that we learned before, with our capillaries, right? We're getting it into and out of the blood. So the respiratory zone is going to be down at our bronchioles, and we'll talk about how there are what are called terminal bronchioles and respiratory bronchioles. But those are smaller branches, and then alveolar ducts, you saw those in the microscope, right? Those are thin walled, and then alveoli. Those are our main areas for gas exchange itself. So yeah, so included in the conducting zone, I should have bronchi in there as well, because those are still big and thick, so we can't do exchanges across their wall. So what we're seeing is this is our kind of full pathway here. And it's not until we get down into the lung itself, into the microscopic structure of the lung, where we're going to be able to exchange gases. So most of the beginning here is just kind of going through those anatomical structures. And we'll do kind of a tour, if you will, follow along as if we were an oxygen molecule, and mention how, as we pass these different structures, what roles they may play. But there's our main layout of it, right? We said it's a pretty compact system, going basically from your nasal cavity up here down to the diaphragm, and that's where it stops. So it's not all over the place per se, right? Okay. So we're going to look at that as we're doing our tour. And some of the larger structures that we were talking about, and some of the things that we've measured so far, has to do with one of 4 processes that we're going to need to understand in order to learn how this system does its job. Okay? So the first one is just ventilation. So ventilation is getting the air to move into and out of the lungs. That's it. So it's not about gas exchange, it's not about gas transport, those things are coming up. It's just getting it down into those alveoli where gas exchange can occur. So how do we do that? So pulmonary refers to the lungs. We're going to see that again and again. Pulmonologist, right, study lung function, etc. And ventilation is this process of getting it in and out of the lungs. That's also just called breathing. So that's the first process. So we're going to need to understand how we do that. Then we've got gas exchange. So when those respiratory gases, oxygen and carbon dioxide, are going to exchange, that's called external respiration. They're exchanging with the gases in the external environment. So we bring the air into the lungs, and then we do a quick exchange. We get oxygen to go from that freshly inhaled air into the bloodstream, and we pull carbon dioxide out the other way from the bloodstream to go into that air in the lungs, where we then go and exhale it into the surrounding environment again, right? So that's called external respiration. So with that process, that second 2 of 4 processes, we need to understand, okay, conceptually, that makes sense. We want oxygen to go into the blood so we can give it to the cells. We want carbon dioxide to come out of it so that we can breathe it into the external environment. But how? How do we get oxygen to go in? How do we get oxygen or carbon dioxide to come out? So it's kind of like when we were in the cardiovascular system, and we got down to those capillaries, and we're like, okay, how? How do we get nutrients and oxygen and fluids to go out? How do we get things to come in, right? So we need to understand those processes behind it. So external respiration, we'll talk about that. Then, once we get oxygen into the blood, we've got to transport it because that exchange is going to occur next to the tissues of the lung, but we've got lots and lots of different cells of the body all around that are going to need that oxygen. They need it distributed to them, and in exchange, they're like, hey, I got all this built up carbon dioxide. Can you take this from me? And then we got to get it all the way back up to the lungs. So the third step is going to be gas transport. And we've already had a little bit of an introspective into that, a little peek into that, because we learned a bit about the blood. So what's the main way that oxygen is transported in the blood? Hemoglobin attached to the hemoglobin molecule in the red blood cells. So we already have a little bit of an understanding about that. So we're just going to build on that a slight bit. So we're a little bit ahead of the game there. Carbon dioxide, we didn't talk about too much in terms of its specific transport mechanism. We do know that once hemoglobin drops off from one of its 4 binding pockets, drops off in oxygen, it now has a free hand. It can pick up carbon dioxide, but that's not the main way that carbon dioxide is transported. The main way is actually in the plasma. So we'll review that as well. And then finally, the fourth step is what's called internal respiration. We did an exchange in the lungs and the pulmonary capillaries at first. We then transported these gases through the cardiovascular system, and then we're going to do an exchange again, an exchange between our systemic capillaries and those cells of the body, like the muscle cells in the bottom of your foot, getting them oxygen and picking up their carbon dioxide. So this is called internal respiration because we're exchanging between 2 internal areas of the body, between the systemic capillaries, the blood in the systemic capillaries, and those tissues. So this is all an internal trade-off or internal exchange instead. Now the good thing about number 4 there, internal respiration, is that it uses the same mechanisms as external respiration. It's just done in a different place. So once we go through external respiration, we talk about how we can get oxygen and carbon dioxide to move across that thin membrane. It's going to do the same process in internal respiration, it's just happening in a different place. So it's kind of a twofer. Once we understand it for 1, we're going to understand it for 2. Okay? So like I said, there's various ways that this should be a little bit easier based on things that we've learned before. But those are 4 key processes. And so in terms of looking forward to exam number 2, that seems like a weird phrase, huh? You're looking forward to exam number 2? Maybe. But that means that each one of those could be like a short answer question. We start looking through the exam objectives. We've got an exam objective that addresses each one of those. So we just need to be able to explain how those 4 processes work. And that gets us to understanding how the respiratory system works as well. Okay? Okay, so those are key. Now, oddly enough, the first two of those are part of the respiratory system. The last two of those are actually part of the cardiovascular or circulatory system, right? But it is important that we understand how that works in order to understand how this system achieves its function overall. All right. So what we see here is a diagrammatic depiction, if you will, of those different processes. So up here, we're seeing inhalation and exhalation, right? So breathing in and out, up here is ventilation. So we need to understand this process of how we get the air to go into and out of those alveoli, those little bubble-like structures that make up the lungs, that make them so spongy and soft, as we were talking about a little bit last time in lab. So ventilation is there. Then we see our second process right here. This is external respiration. So the exchange that we get of oxygen and carbon dioxide between the alveoli and the pulmonary capillaries is external respiration. So that's a very simple definition of external respiration, right? Exchange of those respiratory gases between the pulmonary capillaries and the alveoli. Because the air that's in the alveoli technically is still connected with the external environment. So that counts as an external gas exchange, an external respiration. Next is going to be gas transport. So gas transport is what we're seeing throughout the middle here, right? How is oxygen transported? How is carbon dioxide transported within the blood? And then finally, we have our internal respiration. So the movement of carbon dioxide and oxygen between the systemic capillaries, bless you, and the tissues. Again, now we have a basic definition for that. But we've got to be able to explain it. How and why does oxygen move from the systemic capillary and go into the tissues? Meanwhile, carbon dioxide goes the other way. How do they know, if you will, to go the right way that we want them to? So we're going to learn that as well. All right? Okay. So questions about big picture so far? So putting stars by those processes and starting to understand those processes is what we're doing. Yeah?
So when you're referring to systemic capillaries, you're talking about the cardiovascular blood capillaries.
Yes, yeah. Systemic and pulmonary capillaries are going to be our cardiovascular capillaries. And that's a good, thing to clear up because as we talked about before, we did have lymphatic capillaries as we just finished up that system, right? And those are more about fluid transport, about cleaning those fluids, as we talked about, like with the immune system, etc. So those are going to be, the ones we're talking about here are cardiovascular. Yeah. Mm-hmm. Whether they're cardiovascular associated with the lung tissue or whether they are cardiovascular associated with the body tissues, like out in the fingers and out in the intestine and things like that. Okay. So we'll do a bit of our tour going through different parts of the respiratory system and their role. So this part's not too difficult and just kind of explain why we have these different things, right? So we've got our nose, one of our openings that is going to allow this air to enter in and exit our respiratory system. Along the way, as it's coming in, we're also conditioning this air because when we get down to the alveoli, they are, as we saw in the microscope, very thin-walled structures, very delicate structures, and they need protection. So kind of like we protected the capillaries from the arterioles, right? The arterioles are protecting them in various ways. The passageways that lead down to those alveoli are also, to a certain extent, protecting them, right? So we know they're mucus-lined, as we talked about before. We have cilia there, so we're trapping particles as part of our external defenses and things like that. But also, this mucus-lined pathway and these cavities along the way help to give us a chance to warm the air and moisten the air as well. For us in the desert, there's some harsh air out there, right? So it's very, very dry, and it may be, it's actually pretty nice out there right now, but it may be cold today. The temperature is going to be a bit different than our body temperature. So you know that even if you went up to Flagstaff or something like that, right, we're seeing a cool down at the end of the week. It's going to drop at least 10 degrees or something again, which is okay. We don't want to get too warm too fast, right? All too soon it's going to be too hot. But If you're up in Flagstaff and you're breathing this coming weekend and it's getting really cold, you can feel that in your nasal passages, maybe even the back of your throat, like, right? We don't want that cold, harsh air to reach down in the alveoli as well. So part of the role for the nasal cavity, etc., is to give this air a chance to kind of swirl around a bit. And as it comes into contact with the mucous membranes, we're getting rid of some of those dust particles or pollen or whatever, but also it's warming and it's humidifying it. So that by the time it gets to the back of the throat, hopefully, and then down further, it's been conditioned. So it's this warm, moist air. Now, that is also, interestingly enough, a way that we tend to lose fluid as well. So when we breathe out, and if you're in a cold environment, you can see your breath. Hopefully not because you haven't brushed your teeth, but because it's moistened, right? It's humidified, so you can see the fact that you lose a little bit of that fluid in your body as well. But it's important for us to do that to protect those delicate respiratory passages. So we're filtering, we're warming, we're moistening, we're conditioning this air as it's coming down. And the nasal passage serves as a resonating chamber, so that affects the sound of your voice, whether you're congested and it sounds like this, or whether it's just kind of the shape of that chamber. That also affects how your voice is produced, or how it sounds. And then we have our olfactory receptors, right? Our chemoreceptors in the nasal cavity there too. In terms of anatomy, remember we've got that first room that we call the vestibule. We've got the main chamber, which is going to be our nasal cavity itself. And then we've got our different ridges in there. And those ridges, like I was mentioning, are going to be helping to swirl the air around like that. And so we've got our different types of, sometimes they're called turbinates. But in essence, these are our different conchi, right? Superior and middle and inferior, nasoconchi, and then we have the meatuses, the grooves that are found underneath of them. We work our way to the back of the nasal cavity. We have our pharynx. Remember, we have different sections of the pharynx. We have our nasopharynx, we have our oropharynx, and we have our laryngopharynx as we get down to the larynx itself. We have our different tonsils there. We said a lingual tonsil at the base of the tongue, palatine tonsils associated with the palate, and then our pharyngeal or adenoids on the posterior of the nasopharynx that we have there. We also have our oral cavity, right? We can be mouth breathers as well. So we make sure that we've got different types of protections there. We make sure it's a mucus line pathway, etc. Then as we're working our way down, we're going to be talking about how, when we get to the larynx, that's going to be our exclusive pathway for our air, right? Instead. We also know that we have those paranasal sinuses. So these are, again, mucus-lined cavities within bones that connect to the nasal cavity. So if you get a nasal infection, and it certainly is possible, it can migrate to those different sinuses. So that's why you can get that pain right between your eyes. So there's a frontal one, there's a maxillary one, sinuses, sphenoidal, ethmoidal, etc. Okay, so we're working our way down, back of the throat. We get down to larynx, which generically is called the voice box. But we know it's also this important area where we're routing the air and we're protecting that exclusive air passageway. So the three functions that we have for the larynx: patent airway, switching mechanism to go back and forth because we have that. What's the structure that does that? That floppy elastic structure. Epiglottis, right? Covers the glottis, which is the opening itself as necessary, and then opens up again when you need to breathe or when you need to speak. So we have that switching mechanism, and then finally we have our vocal production that's going on in there. So there's some of our specialized cartilages like we saw. Here are vocal folds. Remember, we have true and false ones that are there, and our vocal folds, or true ones, are attached to our specialized cartilages, or A-shaped arachnoid cartilage, our little niblet of corn that sits on top of it, the corniculate cartilage, and we can see how there's also a section of cartilage embedded within the wall, which is called the cuneiform cartilage, where our epiglottis kind of comes down to meet. We see some of our quote-unquote ligaments. You see a cricothyroid ligament and a thyrohyoid ligament. I don't know why it has it listed as membrane instead, right? So these are all different types of fibrous connective tissues that are holding these special cartilages together. And this is what it looks like as you're looking down the barrel of that glottis, right? So here's the epiglottis. that we see there. So this would come and fold down over the glottis as necessary. And again, you can see here's our corniculate cartilage along here. A little bit of our cuneiform cartilage is forming the seat of where that epiglottis is going to come down, and you can see those vocal folds. So our true ones that we have in here, and the false ones, you can see how they're just kind of a membrane along the side. So when we're looking at our larynx model, you're like, I don't know, they kind of look the same. But when you see them in actual anatomical structure, this isn't going to produce any air. But right here, as the air goes by, like any other wind instrument, as the air travels by something of different sizes, it produces different pitches and things like that, right? So if you have a small piccolo, a small flute, it's going to make much higher pitch sounds as opposed to something like a bassoon, which has a much larger diameter. And that's why we have a difference as well between male and female pitch. That tends to be because in male, part of our secondary sexual characteristics that we'll talk about towards the end is that the larynx tends to get bigger. There's various parts of the body and the musculature that changes, but the larynx gets bigger. And as it gets bigger and it gets a bigger diameter, that means the sounds that it's going to produce are going to be deeper sounds, right? Lower pitch sounds, etc., as opposed to higher pitch sounds. So that's our larynx. Then we get to our trachea, our exclusive air tube. We've seen this in the microscope already. For the most part, we've got three main layers. But remember, one of the key aspects of our trachea that we're recognizing in that tube is that we have that hyaline cartilage, right? Our tracheal rings that are found there in there too. So technically, our C-shaped hyaline cartilage rings count as part of our adventitia. So the innermost one is the mucosa. That's where we're conditioning that air, right? Interacting with it, etc. Submucosa underneath it, where we have our tracheal glands. Then our hyaline cartilage ring and our adventitia towards the outside of it as well. That hyaline cartilage ring is going to be important because later on, well, it's going to be important to keep the air tube open, but it's important later on in lab because seeing that ring is going to help us to identify that trachea slide, as opposed to later on, we're also going to be looking at like the wall of the stomach and the wall of the small intestine, like the duodenum, etc., which also has a mucosa and a submucosa to it. But then it's going to have a muscular layer for squeezing the food along, as we're talking about it in lab, as opposed to an adventitia and a hyaline cartilage layer instead. So seeing that hyaline cartilage was one of the things that will help us to understand we're looking at the trachea as opposed to the muscular esophagus. So here's our anterior, that's the front. There's our hyaline cartilage ring that I was talking about, our submucosa underneath it, and then our mucosal lining on the inside. Remember that the hyaline cartilage ring, we call it a ring, but it's a C-shaped ring, which is connected by that trachealis muscle posteriorly as it faces that muscular esophagus on the posterior side. Okay, and we had talked a bit about respiratory system and how smoking is bad for it, right? Because you have all these contaminants that are accumulating within the lungs, making it thicker along our respiratory membranes where we're trying to exchange, and also it can paralyze those cilia that are part of our pseudostratified cilia columnar epithelium. And so that's helping to warm and moisten the air. That mucus is going to tend to accumulate and settle down into those lower respiratory passages rather than being moved out by those different little cilia there. So, smoking's bad, right? We know that. Okay. So there's our kind of depiction of our bronchial tree and our lungs like we've seen in lab as well. Remember that as we're seeing those branches, it's kind of like an inverted tree, right? So if you flip that upside down, it would definitely look like a branching tree. And a tree does the same thing. We have a trunk, right? The main part, which is going to be our trachea. And then it's going to split. It's going to split into finer and finer branches until you get out to the very tips. And at the very tips, in a tree, what goes on? Gas exchange. So we're getting things like carbon dioxide and oxygen. They are exchanging there, much like we are exchanging down at the delicate branches of the alveolar ducts and the alveoli. All right, so there's actually like 23 orders of branching. Luckily, we don't have to identify all of those, but we do need to know first set of branches, right? The primary, second set of branches. Now, on the left side, we're going to have two secondary bronchi. These are going to the individual lobes, and we only have two lobes on the left side. 3 lobes on the right side. So we have 1, 2, 3 different secondary bronchi on the right side. And then we have tertiary. So tertiary are also called segmental bronchi, and they're going to find finer and finer little subsections of those different lobes. So primary, secondary, tertiaries, that's about where we stop. And then there's continued branching after that. So we're going to go from the bronchi to the bronchioles. And as we get smaller and smaller, not only are we going to get smaller branches, but they're also going to change, right? So the wall is going to change. We start out with cartilaginous rings. They're going to shift to thinner cartilaginous plates. And then the cartilage is going to go away altogether, in fact. Because as we get down further, those bronchioles, much like arterioles, are going to be able to change diameter. So we can constrict and dilate those tubes with smooth muscle, which, just like in the blood vessels, affects flow and pressure, right? So we can constrict, it's called bronchoconstriction, bronchodilation when we do it in the lungs. But if we had all that cartilage there, then it would kind of keep it open all the time. And so we tend to get rid of that cartilage as we get further down. More smooth muscle, less cartilage, and even the epithelium changes. Because we start out with, what's the type of epithelium that we have in the trachea that we saw? That big long name? Yeah, he's going microbiology now, yeah. So it's pseudostratified ciliated columnar epithelium, where it looks like it's more than one cell layer, but technically it's not, right? So it's a little bit thickened. And then we're going to transition to columnar epithelium. So columnar epithelium is going to be a little bit thinner than that. Then we're going to go down to what's our smaller one that's squarish? Simple cuboidal, right? Still simple. And then finally, we're going to get down to the thinnest one, which is going to be simple squamous epithelium. That's what we're going to have down at the alveoli. Just like the capillaries have to be super thin to facilitate that exchange, right? So we're seeing that link with structure and function again. We need a little bit more protection. In fact, when you get to the nasal cavity and the oral cavity, that's going to be more of like our stratified or truly stratified squamous epithelium. Because there's a possibility there's rough stuff going in there, right? Food's going in there, crunching up chips or something like that. I saw some meme recently that was showing, like, it showed a cereal bowl and a spoon, and it had some milk in there, and it had a bunch of thumbtacks in it. And it's like, when you're still really enjoying your Captain Crunch, you really torn up your roof of your mouth, but you're still going back for seconds. Right? So there's rough things that go in there, so we need layers of protection. And so we start out with layers of protection, but we're going to work our way down to thinner and thinner layers that facilitate the function further down. Okay, so we're working our way down from the conducting zone, where it's thick, down to the respiratory zone. So we get down to the respiratory zone, and now it's become thinner. We see that we've lost our cartilage, and now we're down to that smooth muscle layer. These are our terminal bronchioles, right? Some of our last little bits of our conducting zone. And we're transitioning to the respiratory zone where we can do gas exchange. So we go terminal down to respiratory bronchioles. And that term says we're now starting to do exchange across the wall. So respiratory bronchioles, alveolar ducts, alveoli, and if we have a bunch of alveoli together like a bunch of grapes, those are called alveolar sacs. All of those things are now thin enough to allow external respiration to occur, gas exchange to occur on them, okay? And you can see we're working our way down to those structures that are thin enough and numerous enough. About 300 million alveoli. Lots of them, because as we were talking about when we were measuring those lung volumes, you've got like maybe a second or so when you've inhaled air before it's shooting back out again. So we don't have very long to do that gas exchange with the pulmonary capillaries. So we need a lot of area to be able to do it very quickly before that air leaves again. So that gives us a lot of surface area to be able to do that. And lots of surface area with the alveoli, and those are surrounded by our pulmonary capillaries, right? So the pulmonary capillaries are going to be those that are surrounding it, like a web, like you see here. In fact, on the right side there is a scanning electron micrograph showing you that web-like network of the pulmonary capillaries surrounding each one of those alveoli, right? Because that's what we're exchanging with. We're exchanging between the alveoli and those pulmonary capillaries. So this view now is looking at individual alveoli, termed alveolus, right? So the US is singular there. Each one of those are individual alveolus, and we're looking at their structure. And we mentioned that their wall, and we saw it in the microscope, is made up of simple squamous epithelium. That simple squamous epithelium is sometimes here just called a type 1 alveolar cells. It's simple squamous epithelium, but it's called a type 1 alveolar cell. If we have a type 1, it makes sense that we're going to have a type 2. So the second type of alveolar cell is a very interesting one. This one is a surfactant secreting cell. All right, so when we were talking about inflating and deflating the lungs, right, we talked a little bit about how that's like inflating and deflating a balloon, and how the lungs are going to be inflating and deflating. All of these alveoli that make up the lung tissue is going to be inflating and deflating, right? We have that residual volume that keeps these partially inflated so that the walls, the interior walls of the lungs, don't stick together. We know that it's lined with mucus, there's a thin layer of fluid in there, and so just like fluid on the inside of a used balloon, which is kind of disgusting, but little kids do that all the time like we were talking about, right? If you have a used balloon, somebody's blown it up, now it's deflated. If you try to blow it up a second time, you have surface tension of the fluid in there that's holding the walls together. And so it's much harder to inflate it a second time because of that surface tension. We do not want those alveoli fully collapsing and having those walls stick together. Because your very next breath, you'd be like, trying to get them to inflate. Okay? So they deflate partially and inflate, but they never fully collapse. One of the reasons, besides that residual volume, is that surfactant that's made by type 2 cells decreases the surface tension of the little bit of fluid that's in there. Surface tension, right? So if you've ever seen like on a stream or a pond or something like that, there are certain insects, water striders, that can just basically stand on the surface of the water and they don't sink in. They're using that surface tension. Obviously they have a very low mass, but they have a lot of surface area and they're kind of sitting on the top of it like that, floating, if you will. And they are using that surface tension to stay on the surface of the water. So surface tension is where these water molecules will kind of orient next to one another, using the hydrogen bonds, etc., and they kind of stick together. That kind of function that we see, if you will, of water can be bad for our alveoli. So surfactant decreases surface tension, so there's not quite so much pull of the fluids trying to collapse these alveoli. Okay? Now the interesting thing about that as well is that when you hear about babies that are born prematurely, they go into an incubator quite often to finish. One of the reasons they're in there is to increase the oxygen because their respiratory systems are not quite developed yet. One of the last things to develop are those type 2 alveolar cells that are secreting surfactant. They're not quite up and running yet because when the baby's inside, those lungs are effectively filled with fluid, right? So we're going from a fluid environment to a gaseous environment. So there's a transition there. And so if the baby's born early, they don't have that surfactant, enough surfactant, and so those alveoli are having a hard time inflating each time. So we increase the amount of oxygen that's around them, and that aids in that process of gas exchange while their surfactant cells or type 2 cells are still developing and coming online. Okay? Okay. So we see type 1 and type 2 alveolar cells. You can also see that there are some macrophages in there too, right? So we learned about macrophages that are wandering around looking at getting rid of dust or pollen or maybe bacteria that may have gotten down there or something like that, right? So we have some of our immune system on patrol there as well. And so the thin wall of this alveolus with their type 1 cells and the thin wall of the surrounding capillaries make up what's called our respiratory membrane. So that's what we see at the top of this slide. It's talking about the respiratory membrane. So the respiratory membrane is super, super thin. So it's alveolar epithelium, simple squamous epithelium, capillary endothelium, which is basically the same, simple squamous epithelium, and then a fused basement membrane. So the thinner it is, the easier it is for gases to get across. If it's really thick, they got to march. It takes time to get across. We don't got time for that. So we make it as thin as possible. So that's all there is between the areas where these gases need to exchange. So that's going to facilitate that external respiration that we see there. Okay. So right now what we've done is we've worked our way down to the microscopic structures of the lungs in terms of the anatomy. Now we need to just kind of scale out a little bit in terms of other things associated with the lungs. We have the pleurae. If you remember, we have serous membranes on these organs that sit within a cavity. So on the surface of the organ, we're going to have a visceral layer, right? We had a visceral pericardium before. Now we're going to have visceral pleura. We had a parietal pericardium before lining the cavity. Now we're going to have a parietal pleura. That's going to be lining the pleural cavity itself. So those are going to be important in terms of keeping friction down and things like that. But it's also going to be important in terms of, we'll talk about this coming up, keeping the wall of the lung attached to the wall of the cavity. We said inside of the lungs it's important to keep that surface tension down so that things don't stick together. But outside of the lungs, when they're in their cavity, that's actually really important. There's a little bit of space and a little bit of fluid between the surface of the lung and the wall of the pleural cavity. And we want, when we move the wall of the cavity, using some of our respiratory muscles, like our diaphragm, like our internal intercostal muscles, stuff like that, when we move the wall of the cavity, we want the wall of the lung to follow it. We don't want it hanging loose inside the cavity like that. So in fact, there is some surface tension that we're using on the outside of the lungs associated with these serous membranes. We'll talk about that coming up too. Okay. So far, we're just laying out the system. Does that seem okay? Questions? Anything along the way there? One of the things that you would want to be able to do, and you'll see this as exam objective, is you would want to be able to trace or follow a molecule of oxygen as it passes through all of these different parts of the respiratory system. It's just a way, with these different systems where there's a lot of flow and we got to follow things along, respiratory system, digestive system we're flowing through, urinary system we're flowing through, etc. We commonly have these little tracings that we'll do. So it just is a good way of reminding us what are the anatomical parts and how are they connected. Like this leads to that, leads to that. Okay? So if we were going to do this tracing, if we're thinking about breathing in an oxygen molecule, where are we going to start? What's going to be the first part? Mouth and the nose. It kind of depends on how congested you are, right? So we can start with the nose, and if we were doing that, we could say, oh, okay, well, we kind of have that vestibule as we were talking about, and that leads to our nasal cavity, right? And as we go from the nasal cavity, you're going to get to the back of the throat. What's the back of the throat called? Starts with a P. Yeah, starts with a P, sounds like an F, right? So we have the pharynx. So the pharynx, now do remember that the pharynx, and again, that's how we spell it, Y-N-X, rinx, like lynx. We have the pharynx that divides into 3 different sections, right? So what's the first section called? Yeah, so we have the nasopharynx, crossing the nasal cavity. Then we have the oro, crossing the oral cavity. Then we have the laryngo, right, as we're getting down to the larynx. And that tells us that that's what comes next. So we have our larynx, like that. Then after the larynx, now we have an exclusive pathway for air. What's the air tube? The windpipe. Trachea, right? So we have to use the anatomical terms. We have our trachea. Then, as we were talking about, we're going to separate into branches. What are those branch structures called? So we have bronchi, right? So bronchi are the bigger ones, and we had to learn that there were at least 3 different branches that we had to know, right? So we had a primary bronchi, a secondary bronchi, and a tertiary bronchi. I was just kind of listing them shorthand like that. 1, 2, 3. Primary, secondary, tertiary bronchi. Okay? Then, from the bronchi, we go down to what? What are the smaller branches called? Like arteries to arterioles. We have bronchi to? Yeah. So we go bronchi to bronchioles. So we have bronchioles, and as we were seeing in that diagram, we actually have 2 different types of bronchioles. We have terminal bronchioles, as our last part of the conducting system, and then respiratory bronchioles. Like that. Bronchioles. Now from there, now we've gotten into our respiratory zone. Now we have our alveolar ducts. And those lead down to our alveoli. Those are our structures where we're going to primarily doing our gas exchange. I is plural. US is going to be singular, right? Like cacti and cactus. We get down to our alveoli. Now, as we saw on the last slide, we're talking about a respiratory membrane there. So we get the oxygen down to there, but we have to get it into the bloodstream. So technically, when we get to the wall of the alveolus, we're going to have, again, like our alveolar epithelium. And then we have our basement membrane. And then lastly, we have our, we can call it our capillary endothelium, but that's also our simple squamous epithelium, right? And as we then get across the respiratory membrane, now we're in the blood. So what we're seeing with that is basically our tracing through our respiratory passages. So the nose and nasal cavity to the pharynx are 3 different sections, down to the larynx, to our trachea, to our bronchi, 3 different types, primary, secondary, tertiary, to our 2 types of bronchioles, terminal, and then respiratory, because this indicates now we're going to be exchanging gases across the wall, alveolar ducts to the alveoli, and the alveolar wall is going to be made up of our respiratory epithelium, alveolar epithelium, like type 1 cells, if you want to call it that. We learned that's the other name. Basement membrane, the fused basement membrane, that then attaches it to the capillary wall, the capillary endothelium. You can say capillary epithelium if you want. And then we're in the blood. And blood, then we're talking about, again, we're talking about external respiration, gas transport, and stuff like that. Okay? So being able to list those structures in order is a good way to, again, just kind of look at that anatomy and how it all connects together. Also could be a short answer question. Okay? Questions on that so far? Dead silence is a no?
So what part of this is going to be the inspiration? Like, this is the inspiration part, and then expiration, and then the first.
Yes? Yes. So if we were doing inhalation or inspiration, it would be following that sequence. If we were doing exhalation or expiration, which sounds bad, when you're talking about expiration, it's like dead or something you're supposed to throw out, etc. I am definitely that dad where I'm like, it's still good. That's just a suggestion. Come on. Right? You've taken like the salad dressing out the door. You're like, so what? That's like the sell-by date. They know it's going to be in the refrigerator. I just said that this week. You know you're going to have it on the shelf for like another 6 months. That's okay. Doesn't matter if it expired during the summer. Come on. Yeah. You got to hold that over from college days when you can't afford to buy new stuff all the time. Go down to the used food store, get a secondhand food. Yeah. Which, don't forget, right? We have the food bank over there too, free food all the time. Stop by there after class today, get some new food over there. Don't worry, not used. Okay. So this is going to be our pathway for ventilation, which is a process we're going to be talking about, also known as breathing. Okay, so we have to understand breathing. That's the pathway, but how do we get the air to move down and then out again along that pathway? So as we mentioned, inspiration is the same as inhalation, right? So you can call this inhalation. And expiration is the same as exhalation. Those are exchangeable terms like that. So we have to understand how we're going to get that to happen. And so it's going to deal with, we talked about the fact that pressure is going to come into play. And so that's going to deal with 3 different pressures. So we're going to need to know these 3 different pressures. So what we're getting into now is we're basically saying, okay, we're going to be talking about breathing, our first of our 4 processes, and we need to understand how it works. And a key aspect about how breathing works is knowing that these are 3 pressures that are involved. So remember when we were talking about like bulk flow with cardiovascular, right? When we were doing fluid exchange at the capillaries, There are some pressures there, and we're like, oh, okay, we have to understand that, there is blood pressure, hydrostatic pressure, there's osmotic pressure, and you have to explain the roles of those pressures to get these substances to move. So we're doing that again, but these are the three pressures involved here. So we've got atmospheric pressure, intrapulmonary pressure, intrapleural pressure. So atmospheric pressure, that's out here, right? So right now you have one atmosphere sitting on top of you. All of that atmosphere is sitting on top of you. So that's atmospheric pressure. Even though we know that, we can't necessarily feel the oxygen and the carbon dioxide and the nitrogen in this invisible gas around us, it has mass, right? So it's a substance, it has mass, you can actually measure it, even individual atoms have mass and things like that, right? So if you take all of that gas sitting on top of you, that mass in that space, in that volume, has a pressure. That's what we're talking about. We pack certain things into a certain volume, and that gives us pressure. The more things we pack into a certain volume, there's more pressure. Pressure is going to go up. Fewer things in that same volume, or changing the volume, making it bigger or smaller, is going to change that pressure as well. That's atmospheric pressure. So that's what we think about in terms of weather, right? So if you're talking about these days, there's been all the Pineapple Express and all this kind of weird things, right? But usually, if you're talking about weather, it's a low pressure system that moves in that gives you the variable conditions where moisture increases and settles out and things like that. You get precipitation. High pressure systems is usually warmer temperature, sunnier skies, stuff like that. But that has to do with changes in atmospheric pressure. For our purposes, in terms of breathing, we care about it because that's where we're exchanging air with, right? We're exchanging air with the surrounding environment. So atmospheric pressure is where we're getting that fresh oxygen and putting out our carbon dioxide. For the most part, we will assume that atmospheric pressure is about, and we'll see this coming up, about 760 millimeters of mercury, or 1 atmosphere. Now on the right-hand side, you're like, I like the 1 atmosphere, because that makes sense, right? There's 1 atmosphere up there sitting on top of your head. That's the pressure of it, right? And so sometimes you'll see that unit, but you also see that one. We've seen this one already, right? Millimeters of mercury. That's how we measured blood pressure as well, right? So 760 millimeters of mercury is kind of an average value. It will go down, will go up, like we said, with weather and stuff like that. But for the most part, we're going to assume it's about 760. So we have that pressure. Next one, intra. What does intra mean? Inside, within, right? So intrapulmonary pressure, this is the pressure inside of the lungs. This is also sometimes called intraalveolar pressure. The pressure inside of the lungs itself, inside of those alveoli which make up the lungs. And then finally, we have the intrapleural pressure. That's within the pleural cavity itself. We said that there's that space between the lung and the wall of the pleural cavity. There's that little bit of space right there. That is going to be important as well. All right, now for the most part, we're going to be focusing on these two. Just like when we were talking about bulk flow, there are other pressures and things like that. But for the most part, we're going to be talking about atmospheric pressure and intrapulmonary pressure, because that's where we're getting the air to move between, right? We're getting it to move between out here and inside the lung, right? Getting it to move down inside the lung, getting it to move out of the lung. So that's going to be a dynamic process between those two pressures. The other pressure, intrapleural pressure, the importance of that one, the main explanation why we have to know that one, is as we were saying, we need the wall of the lung to stick to the wall of the cavity, right? So it has to stick to the wall of the cavity. So there's a negative pressure, kind of like a suction, partially due to those fluids and stuff like that, that adheres the wall of the lung to the cavity. So that when we're moving the walls of the cavity like this, the lung wall is following it, okay? So that's its main role. The main role of intrapleural pressure, to explain it in this process, is that it adheres or sticks the wall of the lung to the wall of the cavity. And if we know that, then we're good on intrapleural pressure. Okay? Okay. So now let's go to our other ones, atmospheric and intrapulmonary. Now this one shows us diagrammatically in a picture form where those different pressures are going to be, right? Intrapulmonary within the lungs. Intrapleural is in that space between the lungs and the wall of the cavity. And atmospheric out there, okay? And you can see how intrapleural tends to be a bit lower than the pressure in the lungs, creating a little bit of that suction, right? All right, so let's talk about how we're going to get that air to go into now the lungs, because we want to make sure that the lungs are always inflating and deflating. We don't want them to be collapsed like that. So lung collapse is called a pneumothorax, right? Pneumo has to do with air and pressure and things like that, if you will. And so this is where that lung has lost that suction to the wall of the cavity. So that can happen for a variety of reasons. So it can happen in like a car accident or something like that. So if your body is jolted really significantly, it can break it, right? Break that suction, if you will. Much like if you have a glass that has condensation on it and it's stuck to a coaster and you pick it up, you're like, oh, look, surface tension, holding the two together. But if you go, wow, you're right, if you give it a little shake, it'll probably detach, right? So you can get a pneumothorax from that. Luckily, the two lungs are actually in separate pleural cavities. So you can have one detached and the other one still attached and working normally. So that's a good thing. So it can come from that. It can also come from, there's what's called a spontaneous pneumothorax. You're like, what? Just like all of a sudden, it detaches. So that's a weird thing. That is, they don't really have a good explanation from that from my understanding of it. It happens more in males. It happens more in people that are extremely tall. So I'm just thinking, okay, well, right, if you only have a little bit of fluid there, and now we stretch that fluid out, then maybe it's a thinner layer of fluid that's holding it, and it's a bigger organ that's pulling on it, so maybe that's the reason, not sure. But then to be sure, one of the other ways to get a pneumothorax is, getting shivved in the shower. When you're doing that overnight time in jail, yeah, that can be a problem. Because if that happens, right, so if you get stabbed or something like that or punctured, we have this very delicate balance, this little suction that's maintained between the wall of the lung and the wall of the cavity. And if you create a passageway now from the atmospheric pressure, and now it's right in there, now everything balances, right? So just like putting a nail in a tire, and all of a sudden now that pressure changes, now this pressure changes as well. So any type of a puncture like that, now we've lost that suction, and now that lung is just kind of hanging in that cavity like that, right? You got to reestablish that. You got to reestablish that suction again. So they got to reduce that air pressure. They got to remove that air out of there and allow it to adhere to the wall again. Yeah. So once upon a time, there was a movie, and I tell this all the time. I'm sure that should be in more movies. We're talking today a lab about how Finding Nemo teaches us more about the digestive system. But there was a movie a long time ago that was called Three Kings. So it had George Clooney, Ice Cube, and Mark Wahlberg in it. And so they were in combat. And at some point, Mark Wahlberg gets a pneumothorax. So he gets a puncture on one side. And so what they do is they put a valve in there. They actually insert a valve in there, right? Because they're out in the field. Part of the problem with the pneumothorax is, lungs hanging like this, we're moving the wall of the cavity, and lungs still hanging like that. So that's a problem, because now the lung isn't inflating and deflating as well as it should. But in addition to that, part of the problem is you get a little bit of air that goes in there, but now the air goes into the lung, goes whee, and it escapes the lung and goes into the surrounding pleural cavity, right? So now every time you take a breath in, a little bit of air that you inhale to do gas exchange is filling the cavity. And so now the cavity, the pleural cavity, is filling with more air, and now that's pushing on the lung from the outside and actually squeezing the lung from the outside as well, which restricts its ability to inflate and deflate. So what was happening is, every once in a while with this valve, we had a valve like here that, I don't know, they took it off, it wasn't a water bottle, it was something else, right? So they took it there, and he would have to go shh and let some of that air, accumulated air from the pleural cavity, exit so that the lung could fully inflate again. And at a point towards the end of the movie, they get captured, and he's handcuffed. And so as he's continuing to breathe, that lung is getting more and more pressure and it's getting more and more painful. And they finally convince everybody that he's got this valve sticking out here, he needs attention to it. So they allow him to unhandcuff and he goes, and there's this big pressure release as he can breathe again. So in any case, it highlights the fact that these pressures are really important. So that pleural pressure that we were talking about, that we have there in the lung cavity, is important for keeping that wall of the lung attached to the wall of the cavity itself. All right. So beyond that, what we're going to get into next time, which unfortunately we've run out of our time because we're talking about movies and things like that, is we need to explain this dynamic relationship between atmospheric pressure out here and our intrapulmonary pressure, the lung pressure, intraalveolar pressure, and how we get the air to move in and out. So we'll talk about volumes and pressures and things like that, and we'll finish explaining our first process of ventilation, then we'll move on to our other ones. Okay? So, as we finish immune system this week in lecture, that means immune system lecture summaries due this weekend, so don't forget to get that turned in. Don't forget to do your kind of student study group assignment as well. And then, yeah. Oh yeah, so there's a labeling exercise for digestive system. We just are getting into digestive anatomy, but it's a good way to practice with it. This is the stomach, and this is the curvature of it, this is the esophagus, etc. So that's on there too. So things to keep up with as we're moving forward. Oh, I want to know. You do what?